Hi, everybody. This is another one of our videos for the Animal Behavior Research Project Preliminary Data Analysis Pre-Lab, where you're asked to take your group's data and start organizing it into a form that's conducive to doing statistics and graphing that are appropriate for your experimental design. So for this video, we're going to focus on analyzing data if you chose to use scan sampling with interval recording. Now, what we're going to talk about here are a couple issues associated with avoiding pseudo-replication, and then generally how to process all of the data that's gathered using interval scanning or interval sampling techniques to make sure that we've got our data in an organized form that lets us easily calculate averages, standard deviations, variances, etc., based on tank conditions that we're interested in evaluating. So let's think a little bit about the data that we're actually going to be assessing here. This data comes from a student research project where they were investigating the impacts of tank temperature on cricket chirping. So what they were investigating is whether or not the total number of crickets chirping varied between a hotter tank versus a colder tank. So what the students did here was divide out 30-minute observation sessions into 2-minute intervals. And then every 2 minutes, they noted the total number of crickets chirping in the hot versus cold conditions, as you can see here. So the way that the students have organized this data is that they have each group member labeled at the top, and then the the hot tank versus the cold tank laid out here, and then the intervals from 2 to 30. Now, what you'll notice here is that for the different 30-minute observation sessions that they did, they've just stacked those all up here. So we've got 4 different 30-minute observation sessions here, all stacked one on top of the other. Okay, cool. So a couple things that we're going to have to do to get this data well organized. First off, we need to make sure that we get all of our hot observations together and all of our cold observations together rather than grouped out among different group members, right? Because we don't really care about who observed it in the context of doing our analysis. We care about the conditions to determine whether or not they've had some effect on our dependent variable, which is that number of crickets chirping. The other thing that we have to think about, though, is the idea that each individual interval is not an independent sample from the rest of the intervals. So remember, way back at the beginning of the semester in class, we talked about this idea of pseudo-replication, where pseudo-replication occurs when you appear to have a bunch of replicates in your study, but those samples aren't truly independent of one another. That's what you can run into if you have an interval recording experimental design. So let's think about it this way. Let's imagine that we're looking at the data from group member 1, and at minute number 2, they observe 5 individuals chirping in the hot tank and 10 individuals chirping in the cold tank. Then just 2 minutes later, unsurprisingly, they see a pretty similar number of individuals chirping in each one of those tanks. That's because it is very unlikely that the data that they're recording between these two very similarly timed conditions are truly independent from one another. It is very likely that these 4 individuals still chirping at that 4-minute mark are probably just 4 of the original 5 that were chirping in that hot tank. Same deal with our cold tank. We've got 9 individuals chirping. They're probably just 9 out of that original 10. Now, of course, I don't know that for sure, and I doubt that the students know that for sure either, but it is unlikely that these points, because they are so close together in time, are truly independent samples from one another. So what we're going to do to deal with that issue of pseudo-replication is, rather than treat each one of these individual interval observations as an independent data point, what we're going to do is sum up all of those individual interval observations into one session sum that we're going to use for our data points. But before we can do that, the first thing that we want to do is get our data organized according to condition rather than according to group member, and separate out each one of these 30-minute observation sessions as separate independent observation sessions. Okay, so the way that I've got this organized is the same way that I've got the other videos organized as well, if you've watched those, is that on the first page, I've got the unorganized raw data. So this is as the students presented it. On the second tab, that's where we're going to work through the observation process, where we're going to take this data and get it into a format that's usable for analysis. And then the very last tab is where we're going to have our final organized data product that you could easily use for your data analysis. Okay, so what we're going to do first is just copy and paste over all this data. So I'm not going to bother copying and pasting over the group member names because we don't really care about that. So what I'm going to do is just copy this and then stick it over in my organizing process tab. Cool. So the first thing that I want to do is start separating out my observations according to hot versus cold tank conditions. So what I'm going to do first is just go ahead and highlight all of those time intervals, and I'm going to copy them over here. Cool. And now what I'm going to do is pull out all the cold observations. So I'm just highlighting that hot column. Whoops, sorry, clicked on something I didn't mean to. And then I hit Control X. So that's cutting that out and moving it over here. And I'm going to highlight each cold column, hit Control X, or you can right click and then hit cut and copy over all my cold data to a separate section so that I can deal with my cold and hot temperature conditions separately. Then at this point, what I'm going to do is delete these blank spaces because I don't need them. Delete all of these. And now I've got my data divided out according to hot versus cold conditions. But the next thing that I need to do is divide it up into our independent observation sessions. So you can see here, if you look closely at that times, here's a 30-minute session, here's another 30-minute session, and so on and so forth till you get to all of the 30-minute observation sessions. Each one of those 30-minute observation sessions is an independent data point. So I want to make sure that I treat it as such. So what I'm going to do now is, instead of having all these piled up on top of one another, I'm going to separate them all out so that I can make sums for each of those different 30-minute observation periods. So all I'm going to do is just highlight the start of that first new observation interval and then highlight all the data underneath it. At that point, I'm going to cut that out. So I'm going to do Control X or right click and cut. Then what I'm going to do up at the top here is align it with where that other data set is. So now what I've got is two aligned 30-minute observation sessions for our cold conditions. All I need to do now is that same thing to get the other two independent 30-minute data sessions aligned here for my cold conditions. So all I'm doing is cutting each individual 30-minute observation session until I've got all those observation sessions lined up next to one another. At this point, I'm going to go ahead and expand out my cold labels so that every single one of these columns is labeled as cold, so that I can make sure that I am really confident what this data is showing me. I no longer need these individual interval labels because I've got all of that taken care of in my very first column in the cold session. So I'm going to go ahead and delete those now. So I'm going to delete that, and one more, delete that. Boom. So what I've got now is all four of the cold observation sessions for all four of the students in the group. So as you can see here, that means I've got 16 independent data points for my cold tank observations. Each of the four students did 4 half-hour observations. So 4 times 4 gets me 16 total independent cold tank observation sessions. Cool. Now all I need to do is the exact same thing for the hot tanks. So what I'm going to do here is just insert a few extra columns to give myself some room to work with my hot tank observations. And I'm just going to do that a couple times to make sure that I've got a lot of room. And then once I'm done with that, I can get rid of any extra space in the end once I don't need it. Okay, so now all I've got to do is the exact same thing I already did for the cold tank, but with hot. So what I'm going to do is scroll down through here. I see where my first observation interval ends, or my first observation session ends, and I'm just going to highlight all of the other data and then do Control X or right click cut to take that data from the bottom up to the top. Now I just align it so that my 2-minute observation interval through 30-minute observation are all aligned. And what I've got now are 2 aligned 30-minute observation periods. And again, I'm just going to take the rest of that data, cut it, and align it until all of my independent observation intervals are lined up nicely in an organized fashion. Cool. So now what I'm going to do is again extend out that hot label all the way out so that all of my columns are clearly labeled, and get rid of these extra time intervals that I no longer need to have labeled because they are all clearly labeled with that first column A. Okay. Delete. Got all that lined up. And now what I can do is get rid of all this extra blank space and my second now redundant interval label. Okay. So at this point, what I've done is create one column for each hot versus cold tank 30-minute observation period. I've got 16 hot point data columns and I've got 16 cold tank data columns. Now, like we said, these individual intervals, which within a 30-minute observation period, are not independent of one another. So what we want to do, rather than treat each of these values as an independent data point, is instead sum them all up. So they're now representative of one individual independent data point. So what I'm going to do is make another column or another row label called sum. And instead of adding up all this by hand, which sounds horrible, I'm going to get Excel to do all the work for me. So I'm going to tell Excel that I want it to sum up all the values in each column. So all I have to do in the cell immediately below the column is do equal sum parentheses, and then I just highlight all the values I want Excel to add up. Then I end my parentheses and hit enter. And just like that, Excel sums it all up. Now, the really nice thing with Excel is that I don't have to retype that formula for every single column. Instead, all I have to do is hover my little cursor over that green square in the bottom right-hand corner. And now if I just drag it out all the way across my data table, here we go, boom, Excel does all the work for me. And at this point, I have the sum of every single one of those columns. Now, if you're like me, I like to be able to see all of the data more or less all at once, going from top to bottom. So a handy dandy way to convert this data from columns to rows is the transpose function. So to do the transpose function, what you're going to do is select all of your data. Okay, so I've just highlighted all of the data in my column, and then I'm going to copy that data. So I'm going to hit Control-C, or you could right click and then hit copy. What you then do is select an area beneath those rows. That's where you're going to start transposing your data. And again, transposing just means going from rows to columns or from columns to rows. To transpose my copied cells, I go up to paste and click that little down arrow. This symbol right here is the transpose function. When I click that, it converts all my data from rows into columns. Now at this point, I'm in a really good place to get my final organized data. So what I'm going to do is take my data that is now organized according to column. I'm going to highlight all of it and copy it. So either Control-C or right click copy. And I'm going to go over to my organized data tab. What I'm going to do now is paste that data and say, I only want to take over the values. So no equations, I just want the values. And at this point, what I'm ready to do is delete all of those individual, interval columns that I no longer care about. Hit delete. And now what I've got is my tank conditions, hot versus cold, and the sum of all of those individual data points that now represent an individual sample. So at this point, I'm ready to calculate averages, standard deviations, variances, errors, you name it, in my hot versus cold conditions with data that is no longer potentially pseudo-replicated. So hopefully that was a helpful example for you if you did do scan sampling with interval recording. If you want to follow along, like I mentioned, this data table is available for you in the lab resources module in Carmen. So you're welcome to follow along. And as always, this isn't meant to be an exact representation of how your data might look, but hopefully has provided a nice example that you could follow along with and apply to your own individual data. All right, that's all that I've got for you all for now. So I hope this was helpful and good luck with your data analysis. Have a good one.